Hello students, my name is Mirza Jawad Al Hassanan and I welcome you to the lab number 5 of our Bioinformatics 2 practical course. Today we will be studying a new and advanced concept of single nucleotide polymorphism data retrieval and handling. Students, before we dive into the specifics, let's take a moment to understand the context. In Bioinformatics 2 practical course, we explore advanced topics in the field of bioinformatics, focusing on the analysis of genetic variations and their impact on human health and diseases. Lab number 5 serves as a practical application of the concepts we have learned so far. Now, if we talk about the topic of our lab, single nucleotide polymorphisms. Polymorphisms is a generic term that means many shapes. Single nucleotide polymorphism or SNPs are the most common form of genetic variation found in the individual across populations. These variations occurring at a single nucleotide position in the DNA sequence can have significant implications for human health, disease susceptibility, drug response, and more. As bioinformatics researchers, understanding how to retrieve and handle SNPs data is crucial for studying these genetic variations, functional and clinical relevance. It is a DNA sequence variation occurring when a single nucleotide like adenine, cytosine, guanine, or thymine in the gene differs between members of a species. For example, two sequenced DNA fragments from different individuals, as mentioned here, one have sequence AAGGCCTTA and other have AACGCCTTA contains a difference in a single nucleotide at position number 3 where G is replacing with C. In human beings, 99.9% nucleotides are same, whereas remaining 0.1% make person unique in terms of attributes, traits, and characteristics. SNPs are of two major types, coding region SNPs and non-coding region SNPs. If we talk about non-coding region SNPs, which are also known as intergenic or intronic SNPs, are single nucleotide polymorphisms that occur in a region of the genome that do not code for proteins. These SNPs are located in the non-coding region between genes or within introns, which are non-coding sequence within genes. Non-coding region SNPs include intergenic SNPs, intronic SNPs, promoter SNPs, enhancer SNPs, silencer SNPs, microRNA binding site SNPs, and long non-coding RNA SNPs. If we talk about coding region SNPs, which are also known as non-synonymous SNPs, are single nucleotide polymorphisms that occur within the coding region of a gene and result in an amino acid change in the protein sequence. These SNPs can have direct implications for protein structure, function, and potentially influence phenotypic traits. Coding regions are further divided into synonymous and non-synonymous SNPs. Synonymous SNPs, also known as silent SNPs, are single nucleotide polymorphisms that occur within the coding region of a gene that do not result in an amino acid change in the protein sequence. These SNPs typically occur in the third position of a codon, which is known as the vowel position. Non-synonymous SNPs are further divided into missense and nonsense SNPs. Missense SNPs result in a change in a single nucleotide that leads to the substitution of one amino acid with another in the resulting protein sequence. These SNPs can potentially alter protein function or structure depending on the specific amino acid change. Nonsense SNPs introduce a premature stop codon into the coding region of a gene, resulting in the premature termination of protein synthesis. This can lead to the production of truncated and usually non-functional proteins. Some other types of coding region SNPs are transition SNPs, transversion SNPs, and frame shift SNPs. Some SNPs or variants are harmless and only results in the phenotype changes, whereas some SNPs are harmful and cause diseases, hence called mutations. The SNPs occurring frequency ranges from 1 out of 1000 to 1 out of 100 bases in an individual, which is very high frequency. The primary objective of lab number 5 is to make you understand with the necessary skills to retrieve and handle SNPs data effectively. Throughout this lab, we will explore various databases, tools, and techniques commonly employed in bioinformatics research to analyze and interpret SNPs data. By the end of this session, you should be proficient in retrieving SNP data and preparing it for downstream analysis. This lab will enable you to be familiar with the single nucleotide polymorphism databases, and it will also help you to learn about SNPs types, data retrieval and its interpretation, and how to analyze the SNP data. Now let's take a quick look at the structure of today's lab session. We will begin with familiarizing ourselves with popular SNP databases such as DBSNP, Genome-ID, and Ensemble to retrieve SNP information. Now we will explore different file formats used to store SNP data such as VCF or Plink format. We will then move on to the data submission to DBSNP database. This is the main web page of NCBI database. On the main web page of NCBI, on the left side of the home page, you will find a detailed list of resources. In the drop-down menu, you will find a database named SNP. Search box is to search specific data or gene. You can search any gene here in the search bar. Here you can select VHL, a gene, and search here. This gene will be used in this practical lab. By searching the VHL gene in DBSNP database, a built-in database in NCBI, you will be directed to this page, whereas there are three main sections to see carefully. One is the search result, which includes the total number of SNPs mentioned as 7381 here. Second is the main central box containing all the information of each variant or SNP, including identifiers, the RSD value, variant type, allele, position on chromosome, gene part, functional consequences, and many other attributes. Left bar contains all the information regarding clinical significance of SNPs or variants, and functional classes shows the types of variants in the gene. Publication section tells the source of published work on SNPs, and many more information are present in this section. As we have seen that total variants are 7,381. But if we click on the missense under function class, we can see that the number gets reduced to 600, which shows that only 8.13% of the total SNPs are missense. Please note that the selection of the function class depends on the query we have. Here it can be seen that the entry with RSID RS5030722 have all possible SNPs where cytosine is being replaced by adenine, guanine, and thymine in different studies. The SNP data from DBSNP database can be downloaded in three forms as mentioned in the red boxes. First one is file, second one is clipboard, and third one is collections. The most recommended destination is file, in which we can get summary of the data stored by SNP ID. So the best option to download the SNP data is file, summary, and then click on the SNP ID. The downloaded data will be opened in Notepad as shown here. As discussed in slide number 8, you can see the information of each variant SNP, including identifiers, the RSID, variant type, allele position on chromosome, gene part, functional consequences, and many other attributes. We can run a simple Python code to convert desired data in columns to make different values as an input to different tools for SNP analysis. Here we can see that different attributes like RSID, chromosome position, the genome assembly used to the positioning the variants on whole genome, and the nucleotide alteration in blue, red, yellow, green, and brown colors respectively. Genome-ID is a genome aggregation database. Genome-ID is a database that combines whole genome and exome sequencing data from various human populations. It provides information on SNPs including allele frequencies, functional annotations, and population-specific data. This database have three versions, whereas Genome AD V2.1.1 and V3.1.2 have substantially different but overlapping samples compositions and are on different genome builds. Here we search the Genome AD version 2.1.1 and search the VHL gene as an input. By searching genes in Genome AD, we find a table of constraints and variant co-occurrence. The SNPs were categorized in synonymous, missensed, and peel-off with expected and observed SNV's number. Moreover, genome build assembly, ensemble gene ID, transcript ID, region where the VHL gene present, including the external sources used to get such information are mentioned. Next to this information, a graph of per base mean depth of coverage to see that the average number of times a base of a genome and exome is sequenced. Genome AD also shows clinically verified SNPs from the ClinVar database. The results showed a total of 1,186 SNPs extracted from ClinVar database, including pathogenic or likely pathogenic, benign or likely benign, SNPs with uncertain significance and others. We can filter the SNPs based on the desired type we want. Genome AD not only extract the results from other databases but also provide a graphical representation to show variants categories based on its own algorithm. A tabular information of SNPs in a gene is also shown in Genome AD. We can download this data in CSV format and can view it in Microsoft Excel because in Notepad it will not be seen in separate columns as it's comma separated file. The results file will contain more than 50 columns and can't be shown here. Students are advised to follow the instructions, search the database and explore the CSV file downloaded from the Genome AD database. Dear students, we have successfully finished practical number 5 of the course Bioinformatics 2 Practical. Thank you.